My name is Brittany Choate, and I am Director of Programs for Saliva Direct, Inc. And it's my pleasure to welcome you as attendees who are joining us live, as well as those who are going to be joining us via recording of watching this session later on. We're very excited to have three wonderful speakers joining us from Colombia, as well as Italy, and of course, all of you joining us from around the world in your respective locations. A few housekeeping notes before we begin is each speaker will present for about 20 minutes. We'll then have a live Q&A at the end. I'm very excited to introduce our first speaker who will be joining us today. Dr. Adriana Patricia Corredor Figueroa is going to be speaking to us. She is joining us from the ECCI University in Colombia, and her presentation is going to be on bovine leukosis virus, a versatile agent with zoonotic potential. Dr. Adriana, over to you.
Thank you so much for your invitation. My presentation is entitled Bovine Leukemia Virus, a Versatile Agent with Zoonotic Potential. BLV of this virus is a retrovirus from the oncovirus subfamily, Delta retrovirus 2. These RNA codified for structural and regulation proteins that you can see in this picture. This is an illustration for this virus, so very important. GP51 is an envelope protein responsible to cellular adhesion process. It causes most common neoplastic disease in cattle. So from total of animal infected, 60% are asymptomatic, 30% develop persistent lymphocytosis, and 10% develop lymphosarcoma or tumor. Now, the target cell of this virus is B lymphocyte, so the transmission can occur with any liquid or fluid that contain these kind of cells. The prevalence of this virus around the world is very high, up to 80%. In Colombia, we reported before 62% of positivity of this virus in our cattle. Also, we found DNA of this virus in products for human consumption. We report 49% of positivity of GAG gene in milk and in beef for human consumption. Now remember that the natural host of this virus is cattle, but also this virus was reported for other authors in other species with natural infection. In this case, we reported in Colombia that this virus is present in sheep and in buffaloes with natural infection. In this picture, you can see that the same virus that's circulating in our cattle in blue letters is the same virus that's circulating in other species like that, and also in food that I mentioned before. Now I'm going to talk about zoonosis. This is an infection transmitted for animals to humans. And this slide contains key information regarding with the relevance to research in this kind of virus with zoonotic potential. Now, the big question is BLV is a zoonotic virus? First, we know that this virus is present in colostrum, milk, and beef, but we don't know how this virus can move from the animals infected to humans. But we found this virus in humans because we found or have a lot of evidence that this virus is present by antibodies, proteins, and genome. Now, I'm going to share with you some papers regarding with this discussion. This is the first, is about cellular receptor. In this case, we performed this study with both two proteins, AP3D1 from cattle, like cellular receptor, and GP51 from virus, envelope protein that remember that I mentioned before. And we found the most important amino acid that participated in this interaction. And the next question was if the same interaction can occur with this protein in humans. And the answer was yes. both protein AP3D1 in humans are very similar than this protein in cattle. So the big conclusion was that the cellular receptor involved in viral adhesion is this protein in cattle and maybe the same in humans. This is other study. In this opportunity, this author report these proteins acts as cellular receptor for bovine leukemia virus infection, like you can see in this picture. On the other hand, other author reports this study. This is an in vitro study with human cell lines. And in this case, this author tested different cell lines, but also with epithelial cell lines have infected with this virus, like you can see in this picture. Now I want to talk about BLB in humans. This is the first study was performed in USA in 2014, and in this moment they reported DNA of this virus in breast tissues, and 1 year later the same group report that this virus is a risk factor to develop this kind of cancer in women. After, in 2019, the same group performed this study with human healthy, and they found DNA of this virus in 38% of human blood in this population. Australia have this study, and they report similar results. BLB is a risk factor to develop this cancer. Now, in South America, we have some studies. This is in Brazil, and this is other in different places, but both report similar results again. This virus is a risk factor to develop breast cancer in women. Argentina report other study, and in this opportunity also they report 2 important prognosis markers related with cell proliferation in breast cancer. In Colombia, we have 2 studies. This is the first more than 10 years ago, and in this case they found DNA of this virus in breast tissues. And in 2021, we published this study with a Colombian women's population, and we found again this virus is a risk factor to develop breast cancer in Colombian women. Last year, Mendoza and co-workers published this paper with human healthy, and they found 22% of positivity in this population, and also they report that the veterinarians workers have a risk factor to be infected with this virus, maybe by surgical process. Now, our current study, we entitled Detection of Bovine Leukemia Virus in bone marrow samples from patients with B-cell precursor acute lymphoblastic leukemia and healthy controls. This is our preliminary results, and we found high positivity in both groups tested, like you can see. And also, we found that these samples have a low viral load. And the same situation was reported some years ago in cattle. And in this case, for example, these animals cannot infect other animals by this reason. And now in this picture, you can see that again, the same virus that's circulating in Colombian cattle in this number, black numbers, is the same virus that's circulating in food, other species, and also in this population of this study. Okay, now with all of this evidence that I mentioned before, I propose 9 characteristics in order to determine a zoonotic virus. Of course, this is, I want to talk about BLB, but this is the same situation with other virus like that. First, prevalence. It's important to know the prevalence in both species. So in this case, we reported prevalence in cattle in Colombia and also in humans. And same situations was reported in USA. Second one, viral release. We report that DNA, this virus is present in beef and milk for human consumption. So that meaning of presence of this virus. Next one, transmission. In cattle was reported many years ago. This virus can be transmitted with milk, and the same situation with other retrovirus. And the question now is, what about BLB? Is these products the way for transmission? Next one, hosts and vehicles. Remember that the natural host of this virus is cattle, and also we report in humans and other species. And now the question again, if these products maybe are a vehicle for this transmission. Next one, transmission environment. This situation was reported in cattle by many authors, but for example, in Colombia, Ortiz and co-workers reported some risk factor related with transmission in our cattle. And also, I mentioned before this study by Mendoza and co-workers that they proposed that the veterinarians workers have a risk factor to be infected with this virus. Next one, infection dose in cattle was reported for this author in 2016. I mentioned before. And also the question is, what about humans? Next one, cellular receptor. I share with you 2 papers related with these proteins in cattle, so maybe are the same in humans. Next one, phylogenetic evidence. We report this evidence in Colombia with cattle, humans, other species, and also in food. And finally, associated disease. In cattle, we know many years ago, and now in humans, these authors propose that this virus is a risk factor to develop breast cancer in women. Now, with all of this evidence, finally, this question is again, BLB is a zoonotic agent. What do you think? All of these studies was performed with cooperation all of this institution in Colombia, mainly Adriana University. And our current study was performed with cooperation of this institution, mainly Universidad ECCI. Involved Kerlin Ben Núñez, my master student, Jose Cifuentes, statistician, and Jairo Jaime, veterinarian and immunologist. Thank you so much for your attention. It's a pleasure to share our work with you.
Thank you, Dr. Corredor Figueroa. It was lovely to get to hear about the work that you and your team is doing in Colombia. Up next, we're going to transition from Colombia over to Italy, where we have 2 gentlemen who are going to be joining us to present their work. Dr. Luca Giacomelli and Professor Roberto Eggenhoffner are going to be sharing with us today about the evaluation of rapid detection methods for H5N1 virus using biosensors, an AI-based study.
Thank you so much. Thank you, Gary, for your kind invitation. I'm Luca Giacomelli, PhD, and together I'm presenting with my mentor and friend, Professor Roberto Eggenhoffner, from the University of Genoa. And we're going to talk about our study on saliva-based biosensing with some also potential application on public health. Indeed, we will discuss, first we will present a case study of saliva biosensing in winter swimmers. Professor Eggenhoffner is going now to explain us this particular context. Then we will present quite a different application of saliva-based biosensing, which is an AI-based study on the performance of different saliva biosensors for the detection of H5N1 virus. And then lastly, we will broadly discuss about the strategic implications of saliva-based biosensing. Now, Roberto, over to you. I will assist in sharing the slides and scoring them.
Before diving into the data of the slide, let me discuss its scene. Winter swimming is viewed actually as a lifestyle that builds resilience. Participants describe it as energizing, mood enhancing, and also physically beneficial. But we asked if this, which is a self-directed, self-controlled cold immersion, also produce chronic physiological strain, which are not seen directly. To test this, we focused on salivary cortisol, since this is a well-established indicator of the AGP axis activity. We studied 26 winter swimmers, and we discarded a lot of them for various reasons, and compared them to a group of canoeists, athletes that perform controlled activities, and sedentary people controls. Each participant provided 4 saliva samples a day, and we took these samples both in the day of the event, then in the 2 days immediately after. All this for repeated 5 trials to achieve sufficient statistics. Each participant provided these samples, and I would add to the slide that I doubt they agreed to accept to give us blood sampling for the scope of this research. So saliva is a very valid solution to tackle such kind of researchers. Next slide, please. So in this slide, we see on the left, we have a curve of selected cortisol from individual winter swimmers to show that the profile looks like immediately very different from that of a normal circadian rhythm. At evening, as these numbers are saying, the cortisol level does not return to the expected values, low values, and for winter swimmers it remains rather high. This can suggest a form of stress axis dysregulation, which is an altered physiological rhythm that remains invisible without a biological monitoring. That is one of the reasons that makes saliva such a powerful tool, since it allows to pick up signals long before symptoms appear. The next slide, please. What makes the finding we are presenting rather interesting in our opinion is the disconnection between how the people, winter swimmers, felt their conditions and what their bodies were telling us. As said before, all participants were feeling better after cold immersion, but they described also that they have a lot of good mood when no problem in sleep, and so they showed no apparent symptom of stress or any kind of fatigue. But biologically, the situation is very different. Their cortisol profile are flattened and are not recovering, even on rest days. This mismatch is exactly where saliva-based diagnostic show its strength. Okay, please, next slide. So, based on what we learned from physiological studies that I summarized above, we asked ourselves if we could exploit, again, saliva, to apply it to something even more urgent nowadays, in particular, which is the viral biosensing. The viral biosensing, and we had to choose particularly the age 5 and 1 because it has a considerable high zoonotic risk and high case fatality rate, among others. We are discussing a few methods, simulation-based design, because we decided to proceed in this way to test models and practical conditions. So we had to use no pathogen handling, no wet lab, just modeling the sensors under known salivary conditions. The goal is to suggest a fast saliva-based detection tool to be prepared for an epidemic situation that might come in. The next slide. Next slide. Luca,
So
it's up to you.
Yeah. Thank you, Roberto. So actually, thank you, Roberto, for summarizing this. In this particular paper, recently published in Bioinformation, this particular study, we aimed at predicting and comparing the performance of different biosensor platforms for detection of H5N1 in saliva. And actually, we employed an AI-based simulation approach. This is something we particularly like. Roberto is a pioneer of artificial intelligence, I think. And we decided to simulate using artificial intelligence in order to avoid biosafety constraints from the handling of live virus, and at the same time enabling a rapid prototyping and customization of biosensors in order to have a data-driven optimization for biosensor design and deployment. So the input that we gave were viral load, saliva parameters, and the chemical constants for antibodies binding. And we trained our model using published work and published kinetics of viral antibody interaction. Sorry. And also we modeled different architectures for each biosensor. And we compared the biosensor in terms of sensitivity, signal dynamics, and linear detection range. So maybe Roberto, from you here.
Okay. This table summarizes the three biosensors introduced. The first one is a lateral flow test. The second is a field effect transistor-based biosensor. The third one is a quartz crystal microbalance. All three are widely used as sensors in many other fields to control environment and human health. This is the physical principle about which they work. And here are the development of a model that we used to perform the simulation. Models are not discussed, of course, in this talk, but a lot of details are reported in the publication, which has been reported in these previous slides. And in particular, many details are reported in the appendixes. Next slide, please. In the first sensor, which is the well-known one, since people is used to it because of the pregnancy test or the COVID test, more recently. In this case, the analyte is placed on in a window of the apparatus, and it migrates for capillarity flow on the base. And in the conjugate region, it needs some antibodies, which it can attract, and then they meet the test line where antibodies are placed. In case the antibody has been captured by the connection to the analyte, this line will assume a standard color. Further, some antibodies will migrate further, and they meet the control line, where again they can give another signal, which is needed for control. In this case, we worked with the theory of Luca's washboard and the binding equation, and the model the signal on the basis of these detection limits, which are reasonable for the case of concentration of viruses in saliva. Next slide. The next slide is related to a lateral flow platform, which is suggested. This is based on graphene-based biosensors, which has become widely popular recently. But it is a simple device in which a field effect transistor is applied and slightly modified for a functional bioorganization, in which, of course, we have a different signal when molecules are attracted by the biomarker, which control the biological relevance of the virus which has been introduced. So it may be something like a membrane protein or something like that. In this case, the study is completely electrical, and we have a very fast response with respect to the previous device. And we can also point out that this type of sensors are also very fast, but also very sensitive, and their limit of sensibility is down to only 10 particles per milliliters. So it is the most simple, the most accurate sensor that we are presenting. The next slide, please. This sensor is based on a quartz crystal microbalance, which is done by a quartz crystal surrounded by two gold plates that can be functionalized by inserting above them antibodies, which can, of course, capture the analyte to be detected. The model in this case is the Sawyer-B equation, which relates the mass deposited on the sensor to a frequency variation. It is well known that frequency measurements are the most accurate in every field of electronics. And so this is a sensitive measurement, that is related, unfortunately, to an instrument which is not of so simple use as the previous two I have reported. Please, the next slide. Here we have summarized the main properties of the devices we presented. This is the limit of detectability, the response time, which is reasonable for all the three devices proposed. The response from the point of view of physics is different. We come from the optical signal to the electrical signal of transistors and the mechanical response of microbalance. And the use case that comes from is consistent with the above properties. Scalability of a simple lateral flow device is expected very high, moderate for field effect transistors, and for a lab controlled for quartz crystal microbalance. Okay, Luca, please go ahead.
Thank you, Roberto. So, from a broader perspective, we'll show two studies of our group. One tells us how saliva can reveal some hidden physiology, even when winter swimmers say, "No, I'm well. I'm not having any stress or any strain." And also, in line of principle, saliva-based biosensors can support pathogen detection at a very high scale. And what is the advantage for this biosensing is the possibility of having a non-invasive and also self-driven testing. We have unfortunately learned this during the COVID-19 pandemic, how we use the nose swab, but also the saliva swab. And so self-driven testing and non-invasive testing can really have a major role in preventing outbreaks or providing information on the health status. Let me also spend some words on the use of artificial intelligence. Personally speaking, I come from a dry lab approach, while Roberto comes from a wet lab and both a wet lab and dry lab approach. Why we decided to use AI-driven simulations here? First of all, to avoid handling live pathogens in a lab and to carry on different simulations on different biosensors, keeping testing cheaper and powerful at the same time, allowing us to speed up the design and evaluation of different platforms. For sure, we are not saying that AI-based simulations are an alternative to wet lab validation or can replace it. Actually, we feel that this kind of simulation can be a strategic tool to accelerate the process and to guide the process of developing the biosensor more properly, and to provide several information that should tell us the correct route, the correct alternative to be tested in actual experimentation and validation. So what we see as a future perspective for this, we've seen how we can develop non-invasive biosensing platforms for different purposes, and they can be developed only with a cross-disciplinary innovation across different disciplines, physiology, simulation specialist, engineering, and also they can have implications and require the contribution of public health specialists. What are we going to do now? We are going to expand our modeling, our artificial intelligence modeling for the testing of different pathogens. Here we have used H5N1 as a proof of concept, but we are aiming at developing the platforms for other viruses or even multi-panel platforms able to detect different viruses. Again, using this approach, data-driven, using artificial intelligence and computer-based simulation to optimize our research, and then actually researching it using an experimental validation. And to this end, we are looking for cooperation with experts in biosensor engineering, fluid dynamics, clinical and public health partners, and for sure also experts experts in computer science. And to conclude with a message, in our view, Saliva is not just a fluid, it is a message that can give us several messages for public health and public and personal health. Thank you so much again for your invitation. It was a pleasure to share our results with you.
Thank you. Thank you very much to both of you for sharing that information with us. At this time, we're going to move into the live question and answer portion of today's session. Questions that have come through thus far. The first one is to Dr. Corredor Figueroa, if you don't mind. And that is, what kind of partnerships between human and animal testing or research would be most helpful to advance the science you're working on? What would you like to see to have that screening of these kinds of potential zoonotic diseases be better aligned?
Yeah, now in Colombia, we are working with a lot of entities, institutions, public and private institutions. So, for example, I mentioned Becol is a product for vaccines in Colombia. Also, universities and hospitals that, for in case they're in humans, we have samples in this institution. So it's very important to share all experience or learn. Many professionals work together in the same way. So, for example, in this case, we have statisticians, veterinarians, and me, I am a bacteriologist, and yeah.
Yeah, the interdisciplinary and cross-disciplinary
Yes.
Elements of it that was also shared by Roberto and Luca as well was is really great to hear, and that's something that our team is familiar with and tries to advance as well. So I appreciate you calling that out. Thank you.
No, thank you so much.
The next question we have is for Dr. Giacomelli and Professor Eggenhoffner. The question that came through, it ties into what you all were sharing with saliva being this underlying truth teller, and how it's able to reveal things that maybe we as individuals or patients just don't know. And reminds me of a presentation we had yesterday where we were talking about athletes using saliva to detect gut health and cancer and cardiovascular health. It's amazing. But this question was specifically related to that crossover as you're looking for ways of applying this science, you went from looking at winter swimmers to H5N1. What's next is the question. What are the diagnostic targets you're excited about and looking ahead to pursue with this technology?
Roberto, maybe
Professor,
I can see you are replying, but I'm afraid I cannot hear you.
you were on mute there.
Sorry.
Thank you, Professor.
I think that the reason to compare two very different applications would be of interest for a meeting concerning saliva, which is actually the source of many experiments that can be done profitably in the field of biosensors for my own interest, because I am originally a physicist, but it can be extended to human health fairly easily, of course, with the help of other experts. One could try to exploit and to go further and think to biosensors for particular pathologies like the one that come from cancer, but that is rather different from what we were thinking. I know that studies are on progress in this field, and maybe I can participate as well with Luca in collaborations with hospitals or institutions. But that is a very tough problem. Researchers are trying to tackle it, sometimes with some very, very unusual resources, like animals themselves, like dogs, which can exploit volatile molecules, for instance, that comes from some cancers. But biosensors are below the ability of dogs to detect such molecules for now.
For now, for sure. Well, I appreciate you sharing that response, and also thinking about those kinds of partnerships and inviting others to potentially think about ways to collaborate. It's something we really enjoy about hosting this conference, is bringing together different perspectives, and in this particular conference, it being such an international opportunity for us to see where there's some alignment and where maybe there's an opportunity for continuing those conversations with saliva-based cancer diagnostics, maybe incorporating your AI technology and figuring out how we can advance the science in partnership and in collaboration.
Thank you.
Which is a beautiful segue into another question that came through. I think we've got a little bit of time for just one more, and that's thinking of this global impact and these opportunities for cross-disciplinary innovation, international collaboration. From your perspective, what is the top need or the top wish, if you had one, for increasing that interdisciplinary collaboration and the cross-disciplinary innovation? What do you think we need? And Professor Eggenhoffner, I'd invite you to respond to this first.
Of course, I think that the collaboration with clinicians is most wanted in this field. You may have a lot of money, you may have a lot of opportunities, but if you are too far away from the final goal, which is the patient, you are in a position to have poor results from your work. So collaborations are important. International collaborations are even more important. Of course, also meetings are very, very helpful. And I am satisfied when I can participate to work with colleagues of other skills and so.
Absolutely. Thank you. Thank you for that.
Luca has a lot of collaborations and is very active in this field.
Is there anything else you'd like?
He's not saying anything, but he is very active.
Oh, well, we'll put him on the spot, and perhaps, would you mind sharing some thoughts now that your mentor is giving you the floor? You're on mute right now, though.
You're right, I was mute. Thank you, Roberto. And actually, I quite agree with you and with Dr. Adriana and also Brittany, who all of us today stressed the importance of cross-disciplinary assessment, cross-disciplinary cooperation. And this is actually the way we should push for the future among clinicians, specialists of biosensor, experts of artificial intelligence, and engineers. And so different figures are actually required to improve. Personally speaking, I think the field of cancer that Roberto mentioned and early detection of cancer particle, for instance, the circulating DNA can be also sought in saliva, and this can be something that should be explored in the future.
Thank you. Dr. Corredor Figueroa, any thoughts from you about what is needed for increasing international and cross-disciplinary collaborations?
Yes, additionally, also we need more money, of course, and time for research topics. For example, in Colombia, really we need more time to be researching. Also, I think that it's very important to share our language, your science language, with all people, right? With your children, your father, mother, neighbor, I don't know. Any people, all people need to know our language too, because not only in this situation with all the same people, right? Our colleagues, with partners, no, we need to share with all people. It's very important in order to increase the importance of science topics.
Absolutely. Thank you for bringing that back around and really centering it in something we heard earlier from your co-presenters about making sure you keep the patients in mind. It's also important to keep broader populations, making sure that we're being mindful of the entire community that's involved and not just getting so focused on our individual piece of the puzzle, but looking across the whole community for those solutions. Again, thank you all for joining us.